Hi, I am Dr. Jarrett Lindquist, and I am the Dean of the Jindal School of Psychology and Counseling. I'm happy to welcome you back to our latest episode of Minding the Mind. So today we're going to talk about some basic neuroscience techniques. To start out, let's talk a little bit about what we call neuroimaging. Okay? Now, why this is so important? Because it actually allows us to peer inside the human brain. And functional imaging, which we'll talk about, allows us to see changes in the brain over time. So this has really come about over the last 30 to 40 years. The older technique is called positron emission tomography. And the second technique is functional magnetic resonance imaging. Both of these techniques rely on a single principle. Areas of the brain that are active require both energy or sugar as well as oxygen. Okay? So PET, positron emission tomography, is really designed to look at energy consumption in the brain. We use that as a stand-in for activation. So in this case, we have a radioactive particle that we bind to a little bit of sugar. We inject that into the bloodstream, it's taken up into the brain, and then we can actually image those areas of the brain that are most active, i.e. those areas that take up the most amount of sugar. Okay? So in this way, the hotter colors, orange and red, those indicate the areas of the brain that are most active at any moment in time. Now there is a conundrum here. We want to see how the brain changes over time in response to some manipulation. So to do this, we ask the subject to just sit quietly, calm, cool, chill, relaxed, right? We take an image of their brain, we call this baseline. We then introduce some sort of experimental task that we have the subject do, and we subtract out the baseline from that image. What we end up with is something that looks like over here. We call this a difference image, and you can see that there's a lot of activation in the back of the brain. This is an area called the occipital lobe, and this is where our visual cortex resides. So presumably this is a subject who's viewing some sort of picture or other visual stimulus. Okay. Our second imaging technique, again, fMRI, functional magnetic resonance imaging. So in this case, this equipment is designed to detect what we call hemodynamics. That is, areas of the brain where blood with a molecule of oxygen attached, where that flows. And again, the greater the oxygen consumption, the greater the activation of that area of the brain. So just like we saw with PET, we end up with an image where we have hotter colors, reds, yellows, that indicate high levels of activation. And we can then subtract out our baseline image to end up with our difference image, showing us again those changes over time. So over here on the right, I'm actually showing you an image where we're moving from one side of the brain all the way through to the other side. And you can see that different areas of the brain are more or less active. Cooler colors, the blue that you saw here a moment ago, those indicate areas of the brain that are actually less active than normal. That is, it's shown a decrease in activation over time. So this has really been phenomenal because prior to the utilization of this technology, we had no way to peer inside somebody's skull. We had no way of knowing what was actually occurring in the brain itself. Next, I want to talk to you about brain stimulation. Now, there's a historical story behind this. Our understanding of how the brain and its functions are localized really depended a lot on brain stimulation. So a couple of episodes ago, I talked about brain maps, right? How your motor cortex and your somatosensory cortex represent the physical body. Well, we developed these homunculi, these representations, based on this brain stimulation technique. So an important thing to know about brain surgery is the subject is almost always awake. There are no pain receptors in the brain. So the doctor can pass a small amount of electrical current and they simply ask the subject, what did you experience? So did you feel something? Did your leg or arm feel like it needed to move? And again, based on this very careful analyses, they were able to build up these body maps that we talked about previously. Now, a more common and more recent technique is called transcranial magnetic stimulation, TMS. This utilizes a magnetic current to either activate, that is, stimulate different areas of the brain, or controversially, to inhibit or diminish activity in the brain. So in this way, we can target it to different brain regions, and again, we excite it or we inhibit it, and we assess the functional consequences of that on the individual themselves. So again, another useful technique for understanding neural function, but it's also been utilized for the treatment of various disorders. So for instance, what we call repeated transcranial magnetic stimulation. Using this technique multiple times over months or even years is a very effective treatment for major depression. Next, we have neurophysiology and neuropharmacology. Now, there's a lot of ways we can record the neural activity, the electrophysiological activity of the brain. One of the older is called EEG, electroencephalography. So again, I bet many of you have seen pictures of a subject wearing kind of a shower cap, right? And it has these electrodes on it. So those electrodes allow the experimenter to measure neural activity just beneath the skull. And in this way, we can track changes in that neural firing or that neural activity over time. This has been most utilized in studying what we call the wake-sleep cycle. So when you're wide awake, you start to get drowsy, you fall asleep, and then ultimately you go into deep sleep. How does the brain activity change across each of those stages? Well, for instance, we can see in a person who is awake and aroused, they're showing what we call low amplitude, high frequency signaling. Basically what that means is all the different areas of your brain, right? Those processing spoken words, those that are responsible for vision or sight or touch, right? Those that allow you to focus and pay attention. All of those different areas of the brain are doing their own thing. They're all engaged. So they're all operating independent of one another. That's why we have a lot of very high frequency activity. Now, as you begin to relax and get sleepy, ultimately fall asleep and go into deep sleep, there's this very dramatic change. In deep sleep, we have what's called low frequency, high amplitude signaling. Now, rather than each of the areas of your brain acting independently, in fact, during deep sleep, they're all firing synchronously with one another. And by firing, of course, again, we mean action potentials. So in deep sleep, much of your brain is in synchrony one with another. And that's what we see in detail here. So this image shows an actual subject. It might be a little difficult, but the subject's a little bit drowsy. She's starting to fall asleep a little bit. And as we check that, look at the EEG signal, again, we can see these very dramatic changes over time. Next, we have neuropharmacology. This is looking at the neurochemistry of the brain. Now, in an earlier episode, we talked very briefly about the idea that an action potential leads to the release of neurotransmitter from an axon terminal. Okay? So we have neurotransmitter being released, it travels across the synapse, and it binds to receptors on the other side. So in the little cartoon here, we're looking at dopamine. Okay? Dopamine's our neurotransmitter, and it binds to the receptor. This is the way it works in the brain. But we can take in substances through drinking, through eating, or it could be an illicit substance that we smoke or inject, for instance. These various drugs can have very significant effects on the neurochemistry of the brain. So for instance, if we have a drug that inhibits or blocks the ability of dopamine to bind to its receptor, we call this an antagonist, right? It's antagonistic to the effects of the dopamine neurotransmitter. Alternatively, if we have a drug that mimics dopamine and it binds to those receptors, it can actually enhance the overall function or activity of dopamine in the brain. Okay? So just to make this a little bit more specific, let's look at a specific area of the brain. This is called the basal ganglia. Okay? So dopamine release in the basal ganglia is associated with reinforcement or reward, meaning the more you find something rewarding, the more dopamine release that you see. So we can actually track dopamine release in response to your favorite food, to alcohol consumption, to nicotine, to snorting a little bit of cocaine, which is here in yellow. The greatest, the purple, which might be a little hard to see, but it's way up here. Methamphetamine leads to a tremendous amount of dopamine release, and the methamphetamine acts as a dopamine agonist. It exacerbates or enhances the effects of dopamine in the brain. This is one of the reasons, not the only, but one of the reasons why methamphetamine is so highly addictive is because of the tremendous amount of dopamine release associated with it. Finally, I want to talk about genes and genetic analyses. So there's been this long-standing question in psychology, right? Our psychological processes, do they owe their function and ability to our nature, i.e. our genes, or to our nurture, i.e. the environment in which we're raised? So this debate has really gone back and forth for centuries. It used to be framed as an either-or question. We now know that's overly simplistic. It's always a question of nature and nurture. It's a matter of which one plays a greater role. So how do we know? Well, a lot of this work in psychology, which was started back in the 30s, 40s, into the 50s and 60s, they looked at twins, specifically identical twins who share 100% of their genetic makeup versus fraternal twins who share 50% of their genetic makeup. And here's the idea. We can, as psychologists, we can develop what we call a concordance rate. The idea here is fairly simple. If we have identical twins that share 100% of their genes, if one twin suffers from a particular disorder, let's say schizophrenia, we can then ask, what is the chance or probability that the second identical twin suffers from the same disorder? The higher the percentage, the more confident we can be that the genes, which these two identical twins share in common, that the genes play a large role. So when we look at schizophrenia, for instance, schizophrenia has a concordance rate of about 0.8. If one identical twin has schizophrenia, there's an 80% chance that the second identical twin also has it. So by developing a concordance rate for various disorders, again, it gives us insight into this relationship between our genes, i.e. our nature, and the environment in which we're raised, or our nurture. So based on this, we know that some have a very large role. Schizophrenia, bipolar disorder, genes play a very big role. Major depression, concordance rate of about 40 to 60%. So again, both our genes and our environment play similar roles. Now, if we look at twins and what they share based on that genome that they have in common, well, their personality, their intelligence, their abilities, their attitudes, their interests, their fears, the way their brain operates and the way their heart beats, all of these we know are highly influenced based on the genes because we see a lot of similarity between the two identical twins. Finally, let me end with a discussion on what we call chimeras. So this is a Greek term. Basically means to bring together two or more organisms. So beginning maybe 30 years ago, there's really this sort of upsurge in research that looked at the ability in animals, primarily in mice, to either erase a particular gene, we say to knock out a gene, or to add in a new gene. We knock in a new gene. Now that new gene can come from anywhere. It may even come from people. If we put a gene from a person in a mouse, we have now formed a chimera. Now why this is so useful is by using these knockout and knock-in techniques, we can track the organism, the mouse, over development, and we can look at the consequences of, again, deleting or adding in those genes. It's been highly informative for our understanding of the role of our genetics and specific genes in neural function and the development of the brain overall. So with that, I will wrap it up. Thank you very much. I've enjoyed talking with you today, and I look forward to seeing you again.